Hello and welcome back to the channel. Today we'll be discussing one of the most transformative technologies in modern biology, CRISPR. Now, if you've ever wondered how scientists can precisely edit DNA in order to cure diseases, improve crops, or even engineer organisms, CRISPR is the answer. Now let's dive into what this is and how it works. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This groundbreaking technology enables scientists to selectively modify the DNA of living organisms with unprecedented precision. CRISPR relies on mainly two main components. This is the Cas9 protein and the guide RNA, also known as the gRNA. First, let's examine the Cas9 protein. Well, the Cas9 is a specialized enzyme that acts as a pair of molecular scissors capable of cutting both strands of the DNA at specific sites in the genome. This cutting ability is crucial because it allows researchers to remove, insert, or even replace sections of the DNA at precise locations. Without this mechanism, CRISPR would lack the precision that's required for targeted genome editing. The second part is the guide RNA, also known as the gRNA. The gRNA basically serves as a sort of a navigator, directing the Cas9 to that exact sequence in the genome that needs to be edited. The gRNA is composed of mainly two distinct parts. This is the CRISPR RNA, also known as the cRNA. Now, this segment contains a pre-designed 20 nucleotide sequence that essentially matches the target DNA sequence. This ensures that the Cas9 only binds and edits to that intended section of the genome. The cRNA is specific to that DNA region that's being targeted, and it can be customized for different applications. Next is the transactivating CRISPR RNA. This structural component essentially stabilizes the complex by forming a scaffold that binds to the Cas9, enabling the cRNA to guide the protein effectively. Now, in the case of this diagram, this is the RNA that binds to this Cas9 protein here, thus stabilizing this whole complex called the ribonucleoprotein complex, which I'll later explain later on. Now that we've kind of established the key components, let's walk through the process of CRISPR in action. First, the researchers identify a target sequence in the DNA that they want to edit. In the case of this diagram, this is the region that's highlighted in orange. The sequence is then selected carefully based on its role and position. Well, what do I mean by its position? Well, the target sequence must be located near what is a PAM sequence. The PAM sequence stands for protospacer adjacent motif, and it is essentially a short DNA sequence, typically only 2 to 6 base pairs long, located adjacent to the target sequence on the DNA. Now, this is really important because the Cas9 protein requires a PAM sequence to recognize and interact with the DNA and eventually bind to it. In the case of the Cas9 protein, the PAM sequence it's capable of recognizing is NGG, wherein N means any nucleotide, any of the A, T, C's, and G's, and GG means 2 guanine base pairs in a row. Now, the PAM sequence allows for the Cas9 protein to cut the desired section of the DNA and not anywhere else. After that, the diagram is a little funky here, but the Cas9 protein binds to the gRNA to form what is a ribonucleoprotein complex. After that, the Cas9 and the gRNA complex scans the cell's DNA in order to find a sequence that matches the sequence of the gRNA. Now, the complex doesn't just start searching the genomes randomly. It looks for specific PAM sequence, as we've talked about earlier. Once the PAM sequence is found, the complex temporarily unwinds the DNA around it. It then tests whether the guide RNA sequence is complementary to the DNA that's adjacent to the PAM. If it is a perfect or a near perfect fit, then the gRNA locks onto it and prepares to cut it. At this point, the Cas9 activates its cutting domains. The protein mainly consists of 2 nucleus domains. First is the HNH domain, which cleaves the strands of the DNA that is complementary to the guide RNA. Next is the RUVC domain, which cleaves the opposite strand. Well, at this point, we're left with severed DNA strands. What happens next cleverly leverages the cell's natural repair mechanisms in order to introduce targeted genetic changes, and it is really fascinating, but since it's a very complex process, I'll address it in part 2.